Hello everyone, my name is Diana Wilson and thank you so much for coming to watch my presentation today. I'm so excited to show you the project I've been working on for the last 2 years. My thesis study is about the effects of chemicals from textile processing in combination on plankton communities. Pollution comes from many places including runoff, urban waste, and industry. Pipes like the one on the right can be used to dump wastewater from factories into rivers or lakes. I wanted to know how pollution from textile companies that make fire-resistant clothing might affect the organisms in the water they deposit their chemicals into. One way to make fireproof clothing is through the proban process. This technique includes applying a flame retardant like THPC, curing the material with ammonia, and oxidizing it with peroxide. The chemicals end up mixing together in the wastewater. From literature, I was able to find out how these chemicals individually might affect aquatic organisms. However, it is not known what these chemicals did when all of them were combined, and that was the primary focus of my thesis work. In aquatic ecosystems, the impact of chemicals at lower trophic levels seen in the bottom left-hand corner of the graphic can adversely affect those groups at higher trophic levels, like fish, that humans use for food at the top of this food chain. If there is a lack of food supply for fish because the communities of microscopic plants and animals known as phytoplankton and zooplankton are negatively impacted by chemicals, then fish survival and reproduction may be reduced. It is for this reason I focused my experiments on phytoplankton and zooplankton to examine the potential for an ecosystem to respond to textile chemical exposure. Therefore, based on previous research and literature, I knew that peroxide would negatively affect phytoplankton abundance with no direct effect on zooplankton. However, zooplankton have a potential of suffering from a reduced food supply. The ammonium provides nutrients and promotes growth for phytoplankton, but has been known to have toxicity for zooplankton and will likely have a negative impact on the population. The THPC decreases the abundance and health of both the plankton species on its own. Thus, the combination of the THPC and ammonium was cited to have greater negative effects on the zooplankton than the THPC alone. This combination would be negative, but slightly less with the nutrient addition on phytoplankton. Even though I knew the effects of these chemicals, it was still unknown what THPC, ammonium, and peroxide would all do in mixture. I hypothesized that plankton react differently to the individual chemicals versus the chemicals in mixture. I predicted an increase in zooplankton mortality and stress and decreased phytoplankton abundance when exposed to mixtures compared to exposure to the individual chemicals. For the experimental treatments, I tested a spring water control with no chemicals added, each of the 3 textile processing chemicals alone, peroxide, ammonium, and THPC, and every possible mixture combination. The concentrations tested were based on values previously measured in the environment. The zooplankton species tested was Daphnia magna, or the water flea, in the left-hand picture. This species was chosen because they are an aquatic organism that can be found around the world, and there's a lot known about them from the large numbers of studies they have been used in. We used a phytoplankton food source for Daphnia magna, the green microalga chlorella. This can be seen in the right-hand picture. As you might be able to tell from the left-hand photo, this experiment was conducted in a greenhouse to simulate natural light and temperature fluctuations for 7 days. There were 6 replicates per treatment, and each microcosm consisted of a 250 milliliter beaker with 200 milliliters of treatment solution, 5 Daphnia, and enough chlorella to supply the Daphnia with food for the duration of the experiment. After 7 days of exposure, the Daphnia were removed from their microcosms, as you can see me doing in the picture on the right. Then Daphnia mortality was measured visually, and their stress levels were measured using a catalase assay, which measured the concentrations of a stress protein in the Daphnia. Chlorella abundance was measured in 2 ways, by determining their cell density, which is the number of cells in a known volume, and using their photosynthetic pigments, chlorophyll A, concentration. I also measured water quality throughout the experiment to make sure that their physiochemical variables remained within acceptable limits for survival for the study species, and thankfully they all remained at healthy levels. These measurements included conductivity, pH, temperature, and dissolved oxygen. Chlorella cell density was measured using a flow cytometer, which dragged each cell up through a laser that counted it and represented it as a dot on a graph like the one in the bottom right-hand corner. This graph describes the fluorescence of the cells. The chlorophyll A concentrations of the chlorella were measured by extracting the pigments from the cells and measuring their concentration using a fluorometer. ANOVA and Kruskal-Wallis tests both tested the means of our chemical effects to define whether they were significantly different from one another. For our results, there will be several similar-looking graphs that will be representing the different measurements taken. The treatments will be indicated at the bottom with an H for hydrogen peroxide, A for ammonium, and T for THPC. The measurements will be on the side, in this case, average Daphne mortality, and the asterisks mean that those bars are significantly different from the control, and everything else is similar to the control. The organisms we are studying will be shown in the top right-hand corner, and the statistics outputs will be in the bottom right-hand corner. Now, for this graph, after 7 days of exposure to treatments, Daphnia magna mortality reached 95 to 100% in any treatment containing THPC, while mortality in the control, H, A, or H plus A treatments averaged 25% or less. Therefore, it appears that THPC was the cause of the high mortality in Daphnia magna, since there were no differences between treatments where the THPC was alone or mixed with one or both of the other chemicals. In this new graph, we are still looking at Daphnia, and the x-axis is the same, but this time we are looking at catalase activity. Because of the Daphnia mortality on day 7, evidence of stress had been measured after 3 days of exposure to treatments because catalase cannot be measured unless the organism is alive. Catalase, a stress protein, in the T plus H plus A treatment was the only treatment significantly different from the control, meaning the only treatment that chemically stressed the organism was the combination of all the chemicals. For this graph, we are now looking at chlorella, and there are 2 measures of chlorella abundance represented. Cell density in blue is the physical count of the cells, while chlorophyll A is the color of the cells. Typically, these 2 values match like in the control. What was most noticeable in the abundance data was the high cell density in the THPC treatment indicated by the blue asterisks, but the chlorophyll A did not have the same reaction indicating the quality of the cells is low. The THPC mixtures, particularly the T plus A and T plus H plus A treatments, have the opposite effect, where the cell quality is high, but their cell density is low. I could not say for sure whether or not there was an effect from the chemical mixtures or if THPC had a positive effect on chlorella abundance until we compared it to the grazing pressure from the Daphnia, since chlorella is their food source. Here, we are comparing the mortality of the Daphnia on the left with the abundance of the chlorella on the right. From the THPC bars on both graphs, we can see that there are a ton of chlorella due to the lack of grazing pressure since all the Daphnia have died. This same pattern should be true then for the rest of the treatments with THPC. However, the THPC mixtures are different because of the toxicity of the chemical mixtures. From the data, we could see that THPC had the most effect on mortality in Daphnia and also in chlorella abundance when it was in mixtures. The catalase concentrations were affected the most by the addition of peroxide and ammonium. However, these negative chemical effects were intensified in mixtures. These outcomes support my hypothesis that there would be a difference between what each of these chemicals would do by themselves versus in mixture for both of these organisms. My prediction that mixtures would cause increased mortality in zooplankton was only partially supported because only mixtures with THPC saw high mortality. My prediction that mixtures would cause increased stress in zooplankton was only partially supported because the only treatments with significantly higher stress was the T plus H plus A treatment. Lastly, my prediction of mixtures decreasing chlorella abundance was only partially supported as well, since only one of the measures was decreased in each treatment. The effects of these chemicals and mixtures on these lower-level animals raises a concern for the energy transfer to high-level predators in aquatic ecosystems. If this same decrease of health is seen in the environment, then it could cause a detrimental outcome in rivers and lakes where these plankton reside. Thank you all again for coming to my presentation. I would like to acknowledge and thank all of the people who helped me throughout this process, including my mentors, Dr. Cohn and Dr. Lewis, the graduate students and undergraduate students in my lab, including William Hodge, Susanna Forbes, Caitlin Parker, and Michaela Shirley. I would also like to thank the Chandler family for supporting and funding my research, and Georgia Southern University, the Department of Biology, and the university's honors program. All of this was possible thanks to their help, and I'm incredibly grateful for this opportunity. Thank you to everyone watching who is supporting our virtual symposium, and I hope you enjoyed my thesis presentation.